Hey everybody, let's get going on the cell cycle regulation here. So we talked yesterday about mitosis and the cell cycle and how that works, and so today we're going to figure out how that cycle gets controlled or gets regulated. Okay, so how is cell division controlled, and why do people get cancer? That's going to be a byproduct of this thing not being controlled. So um, reminder, several stages, right, that the cell cycle progresses through. You have G1, S, and G2 all in interphase. You have the four mitosis phases and then cytokinesis. So why does that cell divide again? A few reasons, right? Growth, um, replacing damaged or dead cells, and then reproduction. Actually, we don't talk much about it for reproduction because it's not how our cells go through reproduction, right? We have to use meiosis for that, but some organisms do use mitosis. It's just not us. It's more of the single-celled variety, and not all of those, but some of those are single-cell variety and are going to go through mitosis for reproduction. Just know that it's not us. So there are several checkpoints along that cell cycle, okay, and the checkpoints must be passed in order to let the system continue through the cell cycle. So you can see on the diagram there's a checkpoint in G1, there's a checkpoint in G2, and then there is one called the M checkpoint, which can stand for two things. It is during mitosis, and it's also during metaphase of mitosis, is when that M checkpoint happens. So the question down here at the bottom is, what do you think happens to the cell if the checkpoint fails? Like, what if the cell wants to divide, but all of these checks that are going through says, you probably shouldn't divide. What do you think happens to the cell then? Can it go back to being a normal cell, or what would happen? Well, typically what happens is apoptosis. So that is the programmed cell death. That would be a reason why the cell would want to get rid of itself, because it is not going to be able to function like it should. So give me 1 second. Okay, sorry about that. Had a really bad itch. So apoptosis, if the cell cycle gets caught by any of those checkpoints, it's probably going to go through apoptosis. This is a picture of a cell going through apoptosis, where a cell begins to bleb. That word where it kind of like starts, like pieces of it start coming off of the cell. That means it's blebbing, which means it's not going to make it anymore. So if we look closer at the G1 checkpoint, it's going to determine if a cell should replicate its DNA. You don't want to replicate unless you're really ready to go and if you want that done, right? So it's going to roll through G1, and right before replication, they're going to check and see if it should do that or not. So the cell will either enter into the S phase and go ahead and replicate, or if not, it might go into G0. So sometimes cells just get to their adult age, and they're not going to be that young mitotic cell anymore, and they're just going to kind of live out their days in G0. And we say that those cells are in senescence, and that is basically cell retirement, where they're going to live out their days, and they're still going to do normal cell things as long as they can, but they're not dividing anymore. Okay. The G2 checkpoint, so this is after the S phase, right? So what do you think it's going to check for? It's going to check to make sure replication happened correctly. And so super important. The G2 kind of does two things. It looks backwards to see if replication worked properly, and then it also looks forward to see if it has everything it needs for mitosis to divide the cell. Are the proteins that are necessary there and available? And then you have the M checkpoint, which happens during metaphase, and it's going to determine if all the chromosomes are actually attached to the spindle fibers correctly for proper separation, because you always want half of them being pulled to each side. If it was where 3/4 of them were being pulled one way and only 1/4 the other, well, then you're going to have two pretty messed up cells, and that's not going to work very well. So again, metaphase checkpoint, M checkpoint, just to see that everything's attached okay. So how is the cell cycle regulated? So it is under both internal and external controls. There are signals on the inside of the cell and the outside of the cell that can control this. So here are some experiments to kind of show you how this was figured out. So if you look at these experiments here, 1 and 2, and then you look at the results down below, How might these experiments suggest molecules exist internally that tell the cell cycle to progress, to move it from one phase to the other? What tells it to go, from G1 to S and from S to G2? How does it know to do that? Well, they figured out there's some internal signals. So how would these experiments right here in front of you tell you that? So pause the video, think about that for a minute, see if you can kind of come up with some thoughts. But the main reason, if you look at experiment 1, and you're merging two cells, one of them is in S phase and one of them is in G1. And the result is that both cells, when you merge them, excuse me, both cells ended up in S phase and replicating. Okay, and that's probably not too big of a surprise because S comes right after G1. They're kind of in the same spot. They're ready to replicate. So not too big a deal. But look at the next one. You have one that's in M phase. You have one that's in mitosis. You have one that's in G1. And when you merge those, look what happened. They both went into mitosis. But the problem is you can actually see in the one, let me grab my pen. You can actually see the one over here, this guy right here, he only has one chromosome here. Well, this guy you could tell was copied. So you'll especially see it once my red pen goes away that one of them has double and one of them doesn't. Well, that means the one that was in G1, when it merged with mitosis and it tried to start dividing, it has not gone through replication because there's not double the chromosomes, there's just the one. And so that obviously would be a huge problem, but they were just doing this to see, what tells it to go from one thing to the other. And clearly there are some internal signals that are telling those chromosomes what to do and when to do it. And so internal signals definitely play a role. Here are some more internal controls. So we have what's called the mitosis promotion factor. It is a factor that promotes mitosis. So these two things together make the MPF. So one is called CDK, or is abbreviated CDK. It's called cyclin-dependent kinase. Again, if you just use the words, it's a kinase that depends on cyclin. And what do kinases do? You guys learned this first semester. Kinases phosphorylate things. And when you phosphorylate things, generally that's a trigger or a kickstart to make something happen. So if cyclin is present, then the kinase is going to phosphorylate and do its job. And so what's the next thing? Well, the next thing is the cyclin. That's what the CDK depends on, is the cyclin. And that's the on switch for CDK. And so if you have these two things, CDK and cyclin, this is what's going to tell mitosis to go. It's going to promote mitosis. So the MPF is made of these two things, CDK and cyclin, and it's there and it's needed to turn on other proteins as well, such as like microtubule formation, etc. So here's a little diagram that kind of shows you the cyclin and the MPF. So as the cyclin goes up, then the MPF, which is the orange line, follows. And as the cyclin drops, then the MPF drops. So you can tell it's kind of dependent on what the cyclin's doing and whether it will go. And remember, we only have cyclin for a little bit. Oh, let's get rid of that. We only have cyclin for a little bit. Sorry, a lot of pop-up windows. We only have cyclin during mitosis. After that, it goes away, and we'll talk about that in a second. Actually, we'll talk about it right here. So another diagram here. So you see G1, S, and G2.